Hello, my name is Dr. Roderick L. Roll, and today I will talk about uh, chemicals that affect microbial growth by binding and inhibiting DNA or RNA synthesis. So, inhibition of DNA or RNA synthesis can occur through consuming these following drugs, and we'll talk about all of them. The first drug we'll look at is actinomycin. So previously, you talked about DNA replication in your general biology course or your human systems course. And this process involved over a dozen enzymes. And some of those enzymes were DNA helicase, uh, uh, primase, um, uh, DNA polymerase 3, DNA polymerase 1, DNA ligase, topoisomerase. There was many different enzymes you learned. Even you talked about some in for transcription and translation. So think about those enzymes. Now, understand that this drug, actinomycin, is going to bind to the three-dimensional structure of DNA. So it kind of intercalates, it embeds itself. So look at the picture to the right and you can see actinomycin binding to the 3D structure. If it does that, if it's embedded within this structure, how can helicase travel down this three-dimensional structure? How can DNA polymerase 3 read from 3 to 5 if it gets stuck because of this embedded drug? So that's how it works. So DNA replication in prokaryotes and eukaryotes are very similar. Therefore, this drug will indiscriminately affect DNA synthesis in both prokaryotes and eukaryotes. Therefore, this drug isn't normally used because of its non-specificity. Again, if a patient takes actinomycin for a streptococcus infection of the throat, it's going to affect the streptococcus and the patient itself. So both will have inhibition of DNA synthesis. So this drug is going to be used for research purposes and cancer treatment exclusively. The next drug, these are semi-synthetic, these are synthetic analogs. These drugs are quinolones and fluoroquinolones. They're going to target prokaryotic DNA gyrase. Now, in human systems and general biology, I talk about DNA gyrase as topoisomerase. It's the same thing. And how topoisomerase worked is imagine a zipper being unzipped. And as you're as you're unzipping the zipper, if you go upstream, if you go towards the right in this image, imagine a knot uh, forming in the portion that has not been unzipped yet. If that knot forms, you have to be able to unknot, take the knot out, or the zipper will stop. So if the zipper is moving and the zipper is helicase, and then all of those enzymes are creating um, the actual leading and lagging strand. If you cannot break open this knot, you stop DNA replication. What breaks open the knot is topoisomerase. So the quinolones and fluoroquinolones are going to target topoisomerase and inhibit it from working, therefore stopping DNA replication for that bacteria. An example of a drug you might hear about is ciprofloxacin. Ciprofloxacin is a fluoroquinolone. It will target topoisomerase and kill the bacteria by not allowing it to replicate its DNA. Some quinolone drugs can cause positive opiate results with certain brands of immunoassays. As I've mentioned before, I am also a clinical lab director where my toxicology labs look at urine, blood, and feces to determine if patients are using drugs. Sometimes we have false positives for opiates, meaning the patient is not on an opiate drug like fentanyl or morphine. They're not on the drug, but they're positive for that drug in their system. What happened is these individuals are taking ciprofloxacin. So Cipro is FDA approved for treating urinary tract infection, sexually transmitted infections, skin, bone, joint infections, and so on. So if one of your patients is taking Cipro, they would test positive for abusing opiates. Just be mindful of that as a healthcare provider. The next category is attachment nucleotide analogs. They're going to incorporate within the pathogen's genome, which would distort its three-dimensional shape. So if the DNA structure is altered, then those enzymes I mentioned before may not be able to travel and do their thing, which is to replicate that genetic material. This interference does not just affect DNA replication, it can affect transcription and translation as well. Some examples of these nucleotide analogs are in the picture that I've provided. Let's look at this picture in much more detail and look at the center where it says nucleosides. These are variations of a nucleotide, a term that you probably heard of before. So these nucleosides are adenosine, uh, guanosine, cytidine, and thymidine. If you look, I have arrows leading from each one of those that illustrate analogs for each one of those nucleosides. Again, if you don't understand what a nucleoside is, to the right image we have a nucleotide, and to the left we have a nucleoside. The only difference is the nucleoside doesn't have a phosphate. So it's the same thing that you learned before, just remove the phosphate group. Remembering these nucleosides must be incorporated, right? So as you are replicating your DNA, you must have these nucleotides and nucleosides available in order to continue the synthesis of nucleotides, of the DNA. So viruses are more susceptible to these attachment analogs. Therefore, we use them to kill or to destroy viruses, to not allow viruses to replicate. Gertrude Elion won the Nobel Prize in Medicine in 1988. And how this young lady won the Nobel Prize is by creating drugs that act in the manner I just mentioned. So let's look at the first one, ACV. So this is an antiviral drug, the first one, that actually inhibited viruses from being able to replicate. And that's what she won the Nobel Prize for. She also created a antiviral called AZT that did the same thing for HIV. So notice if the patient has HIV and this virus is infecting one of your cells, and what it would do is it would take your nucleotides and nucleosides and create more HIV virus. But if this patient is taking AZT, when the virus goes to replicate, instead of it taking thymidine, it would take AZT. And this would distort the structure of its nucleic acid. And now this virus will not be able to replicate anymore. So that is how the drug works. The next category is rapamycin. So it has bactericidal killing capability, unlike the previous one that affects viruses. So this one, rapamycin, was discovered in 1959, and it is a mixture of 5 substances. So it gives a 1, 2, 3, 4, 5 punch to kill its target. How it destroys its targeted cell is rapamycin binds to RNA polymerase. Remember, RNA polymerase makes a messenger RNA. So if it binds to messenger RNA and distorts the shape, then it's going to inhibit transcription. So we talked about drugs that affect viruses, and we talked about drugs that affect bacteria. So antibacterial agents are called antibiotics. And these antibiotics will not affect viruses such as cold and flu. So we'll talk about cold and flu viruses much later, like the rhinovirus, that's like the cold, and then influenza, that's the flu. So antibiotics will not work on those. You have to take something like a antimicrobial analog. So I mentioned the nucleosides previously and those different versions of them, those analogs, those would be used for viruses, whereas antibiotics would be used for bacteria. So antimicrobials known as attachment antagonists are used for viral infections.